Welcome back to Microbiology. Today we're going to start Chapter 12, Modern Applications of Microbial Genetics. Chapter 12 connects everything we've learned about DNA and microbes to real-world biotechnology, the tools that allow us to modify genes, produce new medicines, and even improve agriculture. And in this video, we're going to begin with the first section, 12.1, Microbes and the Tools of Genetic Engineering. And by the end of this section, you should be able to identify tools of molecular genetics derived from microorganisms, describe how recombinant DNA molecules are created, explain how DNA is introduced into both bacterial and eukaryotic cells, and recognize what genomic and cDNA libraries are used for. Let's start with the big picture. What is biotechnology? So biotechnology is the use of living systems to benefit humans. And modern biotechnology focuses on genetic engineering. This means that we're changing DNA sequences to make recombinant DNA. Recombinant DNA is DNA that combines genes from different organisms. When that DNA is expressed in another species, we call that organism a transgenic organism. A classic example is E. coli that's been engineered to produce human insulin, replacing the older method that used to use pig insulin. Recombinant DNA is created in vitro. So this is outside of the body, outside of an organism, and taking place in like a test tube or a culture dish, anywhere outside of the living organism. And it's introduced into a host cell. This allows us to study or produce specific proteins or traits, and it builds upon natural bacterial processes that we saw in the previous chapter, horizontal gene transfer such as conjugation, transduction, or transformation. So by harnessing these, scientists can insert human genes into microbes, and as I mentioned in the previous slide, we can get transgenic E. coli, where E. coli is carrying genes from humans, and E. coli is going to produce insulin for us. So before biotechnology, before we had the capability to do so, humans, such as patients with type 1 diabetes, would inject themselves with pig insulin because pig insulin happens to be very similar to human insulin. There's only one amino acid difference. So it worked pretty well, and it was the most common source back then. And this figure shows that concept visually. So we have a human insulin gene taken from a human cell, and it's inserted into a bacterial plasmid. You can see we've created this recombinant DNA where we have DNA from two different species, human and bacterial. And that plasmid is then used to transform bacteria. Remember, transformation is when bacteria take up the DNA from the environment, and that gives them, the bacteria, the ability to now produce the insulin protein. So all of these cells, as they divide, will be producing insulin protein. And this is the basic workflow behind most gene-based biotechnology. Molecular cloning is a lab method that builds recombinant DNA and inserts it into a host. So that's basically the summary photo that I just showed you in the previous slide. It uses microbial enzymes and our plasmid vectors from bacterial cells. And the main steps are cutting DNA, inserting it into a plasmid, and then transforming that plasmid into a host cell. The goal is to replicate or express those genes of interest, the ones that we've inserted into the plasmid. And this process was first demonstrated by Boyer and Cohen in 1973. They actually took some frog DNA and cloned it into E. coli. This was a really big or major milestone back then in genetic engineering. Restriction enzymes are the molecular scissors that make cloning possible. They cut DNA at specific palindromic sequences. So remember, palindromes are, in words, when we're looking at words, they're the same word forwards and backwards. So mom, for example, is the same if you go this way or that way. Another common example is race car, right? If you spell it this way or if you're going backwards, it's spelled the same. And when they cut DNA at specific palindromic sequences, these restriction enzymes will either create sticky ends or blunt ends. Sticky ends will have exposed bases that can hydrogen bond with their complementary sequences, and blunt ends will not have any exposed bases. After cutting, DNA ligase, we've seen that one before, is used to seal the sugar phosphate backbone, forming a continuous DNA strand. And together, these enzymes allow scientists to create recombinant molecules with precision. And here we can see an example of how restriction enzymes work. Some restriction enzymes will generate sticky ends and others will generate blunt ends. And here are the names of these specific restriction enzymes. Each will bind to and cut at specific sequences. So BAMH1, these are named after the bacteria they were discovered in. If I look at this first one, the top picture, if I just look at the sequence where BAMH1 will cut, I can see that it's a palindrome because the sequence going down this way from 3 to 5 is CCTAGG, and I see the same exact thing going this way, CCTAGG. So these make up a palindrome. And it just so happens that BAMH1 happens to cut between these two G's across and then over here. After the cut has finished, you can see these are sticky ends because they are no longer paired. They're now unpaired, and they will bind to their complementary sequence. So if you had kept them pretty close together like this, they would just re-stick together, re-anneal. Here's our second one. This is HA3, so also named after the bacteria they come from, Haemophilus bacteria. This one happens to cut right in the center of this palindromic sequence. It's CCGG, CCGG, running down one end or the other. They cut right in the center, so there are no overhangs. There's no sticky ends, and we call this a blunt cut. Plasmids are one of our most common vectors that carry the DNA we're interested in studying. So plasmids are small circular DNA molecules that replicate independently in bacteria. So they usually serve as these vectors, carrying carriers that deliver the foreign DNA into the host cell. And we have several different types of plasmids that are used very frequently in biotechnology, such as PUC19, so plasmid UC19. These include several key features, such as an origin of replication, where they're going to start copying the plasmid. They usually have some kind of antibiotic resistance gene. Here it looks like this one is resistant to ampicillin, that antibiotic. We usually have a multiple cloning site. So this is the multiple cloning site for PUC19. It contains many different restriction enzyme cut points to allow us to select for any of these enzymes to open up the DNA and insert our DNA of interest. And usually it also contains a reporter gene like LAC-Z to screen whether or not we have successfully inserted our gene of interest. Here's another image of that PUC19 plasmid. Again, little p stands for plasmid. UC in this case stands for the University of California, where this was created. So this was UC San Diego, actually. And I like this one because it has a clearer image of my LAC-Z reporter gene, and it shows me where those multiple cloning site with restriction enzymes were located. If I were to cut open my reporter gene, I can see that I would disrupt the LAC-Z gene if insertion happens. And that means this enzyme that's encoded by this gene would no longer be made. And that LAC-Z gene I just talked about is important in something known as blue-white screening. Blue-white screening is used to help us identify whether or not cells contain recombinant plasmids. So it turns out that when that LAC-Z gene is intact, it has not been interrupted, then it normally encodes an enzyme called beta-galactosidase. This enzyme is useful in breaking down lactose, but it can also break down a substrate we often use in the lab called XGAL. XGAL is normally white or colorless, and when it's broken down by beta-galactosidase, it creates a blue product. So when the gene is intact, we will see blue colonies. But if that LAC-Z gene, remember that gene was over here somewhere, if we actually successfully open it up and insert one of our genes of interest inside there, it'll be disrupted. That means we're no longer going to have beta-galactosidase, we're no longer going to digest XGAL, and the color will remain white or colorless. So that means that the gene has been inserted. We have disrupted the LAC-Z gene. So these white colonies will later be selected for further analysis. We usually also sequence the DNA to make sure we've successfully cloned in our gene. And again, this confirms successful gene insertion if we see these white colonies. Let's take a look at what blue-white screening looks like in the plasmid and in the cell. So here's my vector, and here let's say this is that LAC-Z gene. Here's the DNA of interest that I'm trying to insert into the plasmid. There are 3 potential ways it could be inserted or not inserted. If the gene is not successfully inserted, let's say I cut open the plasmid, but it re-anneals or re-sticks to itself right away, there's no insertion, that means the LAC-Z gene would produce beta-galactosidase. It would digest XGAL, producing that blue color. So that's not what we want. We don't want that. Or if the the gene is inserted somewhere else, outside of where I intended it to be inserted, the LAC-Z gene would still be intact, producing beta-galactosidase and digesting XGAL. I would see blue colonies again. That's also not what I want. In the very top scenario, I can see the gene has been successfully inserted within that LAC-Z gene. That means I will not make any beta-galactosidase. I won't be able to digest XGAL, and XGAL, when it's intact, is white. So these cells are the ones I would want. I would pick the white colonies, purify the DNA, and then sequence it to confirm the correct insert. So how do we get DNA into the bacterial cells? Turns out that we usually use transformation. So transformation is the main process of getting recombinant DNA into our bacterial cells. Remember that some species are naturally competent. They can take up DNA directly from their environment. But other species have to be made competent, either through chemical treatments. So in the previous lecture, we talked about calcium chloride, or we can give them electroporation, kind of a little electrical shock. Both of these will temporarily open up the cell membrane so they can take up that DNA. Then the transformed cells are cells that have taken up that recombinant DNA, are selected for by growing them on antibiotic plates. Remember that, let's say this is a cell. We have our normal chromosome, and then this is a cell that has the normal chromosome, but also the plasmid. Remember, on the plasmid, we have some kind of antibiotic resistance gene, such as gene that makes it resistant to maybe ampicillin, that antibiotic. So if we were to grow these bacteria on plates with antibiotic ampicillin, these bacteria would survive, but these would not. So doing this, growing the cells on antibiotic plates, will select for the bacteria that have successfully taken up that plasmid. Then we can go through a blue-white screening to make sure that that plasmid has the gene of interest inserted in the correct region. Quick checkpoint question in blue-white screening: what does a blue colony mean and why is it blue? So remember, a blue colony means that the plasmid's LAC-Z gene is intact and no foreign DNA was inserted into that polylinker site, that multiple cloning site with all of those restriction sites. And it appears blue because the beta-galactosidase enzyme encoded by that LAC-Z gene breaks down the colorless substrate XGAL into that blue product. Whereas if we have white colonies, that means we lack functional beta-galactosidase because we've successfully inserted the DNA into that LAC-Z gene and we've disrupted the LAC-Z gene. Besides transformation, we have those 2 other natural processes that can move recombinant DNA into cells. We have conjugation. Remember, conjugation uses a pilus to transfer DNA directly from one bacterial cell to another through that F plasmid. So we can create recombinant F plasmids and spread them between cells through conjugation. We also have that transduction. Remember, transduction uses those bacteriophages to deliver recombinant DNA. So bacteriophages we saw in previous chapters, carry DNA between bacteria. So we can create engineered hybrids called phagemids, and these combine plasmid and phage sequences, enabling efficient packaging into phage particles. Once they're delivered, the DNA may integrate into the host genome. This occurs during that lysogenic cycle, also known as lysogeny, or remain as a plasmid. So let's summarize molecular cloning using our figure from our book, this molecular cloning flowchart. I'm going to zoom in on the next slide. And here we've zoomed in on the top half of the figure. It begins with isolating the gene of interest, cutting both the gene and the plasmid with the same restriction enzyme. We want to use the same restriction enzyme to make sure they have the same sticky ends. So if I look closely, the sticky ends from my gene of interest is going to be complementary to the sticky ends that I've cut open at my restriction site. And that happens when you, again, use the same restriction enzyme. Then I'm going to ligate them together, right? So ligase is used to ligate the gene of interest with the vector. And now we've created our recombinant plasmids. We're then going to introduce them into bacterial cells. So that's the next slide. Remember, to get the plasmid, these recombinant plasmids, into the cells, we use the process of transformation. And notice that the plasmids have that LAC-Z gene. So we have that reporter gene, and we have an antibiotic resistance gene here. So when we grow our bacteria on our plates, if the bacterium did not take up the plasmid at all, it would not have the ampicillin resistance gene. So if you have a petri dish with ampicillin in it, these cells will get killed off. Now we have these two cells left, right? These two. Looks like both of them have a plasmid in them, so cells will grow. But looks like this one, I see that the LAC-Z gene is intact. Remember, the LAC-Z gene, when it's intact, it'll generate blue colonies, because that means the LAC-Z gene creates beta-galactosidase. That beta-galactosidase enzyme chops up X-gal, forming that blue product. So I don't want the blue colonies. That tells me I have not successfully inserted my gene of interest. But these cells have that ampicillin gene, so they will grow, and they've interrupted my reporter gene. So there is no longer a good or intact LAC-Z gene. That means I'm not going to make beta-galactosidase, right? There's no enzyme. So the X-gal, which is colorless or white, stays intact, and I'll have those white colonies. So I want those white colonies, and then I can either study the colonies further or screen, or I could sequence the DNA to make sure I've successfully cloned the correct gene. Our next checkpoint question asks, what's the original function of a restriction enzyme? So restriction enzymes are actually bacterial defense proteins that cut and destroy foreign DNA. And this was usually to protect them from bacteriophages. So they would chop up the bacteriophage DNA before it had a chance to kill the bacterial cell. In biotechnology, we can take advantage of these enzymes, and we've repurposed them to cut DNA at specific recognition sites for molecular cloning. In the next question, it asks, what two processes are exploited to get recombinant DNA into a bacterial host cell? So we exploit the process of transformation, right? We can uptake free plasmid DNA, and we either have naturally competent bacterial cells or we make them artificially competent through chemicals or electroporation. We can also use the processes of conjugation or transduction, right? Transferring recombinant DNA through cell-to-cell contact through that F plasmid, that's conjugation, or through bacteriophage infection if we're talking about transduction. In the last question they ask, distinguish the uses of an antibiotic resistance gene and a reporter gene in a plasmid vector. So the antibiotic resistance gene, like that AMP we saw earlier, provides a selectable marker. Only cells that have received the plasmid will survive on an antibiotic plate. Whereas the reporter gene provides a visual marker, like that LAC-Z, to show whether the foreign DNA was inserted correctly into the plasmid, and we saw that in that blue-white screening. In the past, by using these molecular cloning methods, we were able to create genomic libraries. And genomic libraries are a collection of clones that together contain all fragments of an organism's DNA. So the genome was cut out with restriction enzymes and inserted into plasmids or phage vectors, and each colony or plaque contains one DNA fragment. So researchers used to screen these libraries to find specific genes of interest, although this is now really historical. Nobody really creates or uses or makes these genomic libraries anymore, because with today's technology, These techniques, the genomic library, has really been replaced by something called whole genome sequencing. But genomic libraries are still important in terms of history, and they're historically important for gene discovery. But a little bit about whole genome sequencing down here. This is a lab procedure that determines the complete order of DNA bases in an organism's genome. And what we do is we extract the DNA, break it down into smaller fragments, and then we sequence these fragments. And these days, technology is so great that we go through a process called next generation sequencing, and we can sequence the entire genome of an organism within a day. But let's look at what we used to have. This is historical. These are our old genome libraries, and we used to take DNA from an organism, cut them up into fragments, and have each fragment inserted into a plasmid vector. Then each plasmid was inserted or transformed into bacterial cells. So we had this library that together made up the genome of the organism of interest. But we weren't limited to just bacterial cells either. You can also make a phage library genome of interest. So same thing, you would take your genome of interest and chop it up with enzymes. Make sure you digest the phage genome with the same restriction enzymes so they have the same sticky ends. And then you would build recombinant phage with your genes of interest. Grow these phage on a bacterial lawn because remember, phage are not alive, they need host cells. And when they lyse the bacteria during the lytic cycle, they generate plaques. Within each plaque, you know, there are thousands or tens or hundreds of thousands of the phage carrying that part of the genome. So collectively, you have all of these phage with that genome of interest. There is something we have to take into account, however, because remember in eukaryotes, when we make messenger RNA, we first make pre-mRNA, and that includes exons and introns. So if when we're cloning eukaryotic DNA, we don't want those introns, right? We don't want those non-coding regions because they wouldn't make sense when something like a bacterial cell is trying to express those genes. Bacterial cells cannot cut out introns. So in order to make a real genomic library that's useful, we have to use complementary DNA, also known as cDNA. So our libraries actually contain cDNA and not regular DNA before the introns have been removed. So how do you make cDNA libraries? We actually use messenger RNA and take advantage of an enzyme that was discovered in viruses, reverse transcriptase. Remember that we find these enzymes in retroviruses. Retroviruses contain reverse transcriptase to read RNA and produce DNA. It's like the reverse of transcription. So this is great for us because when we take messenger RNA that does not have introns, those introns have already been spliced out, and we make DNA, that's complementary DNA because it does not have any introns. There's no non-coding DNA. Now if we insert the cDNA into a bacterial cell, they can express these genes, and the genes, those proteins, those resulting proteins, will be functional. They won't have those introns. So this is very useful for studying gene expression, looking at protein coding regions, and it allows us to clone eukaryotic genes in prokaryotes that are unable to splice out those introns. So here's kind of a more accurate look at our historical genome libraries. We don't actually take DNA, but we take RNA from the organism, and then we reverse transcribe this RNA to make cDNA. So the fragments that are inserted into these plasmid vectors are actually cDNA fragments. That way, bacteria can directly transcribe and translate those genes to produce proteins if we're looking at gene expression. Otherwise, we'll create a library with each plasmid carrying a specific part of the genome, and with all of our bacteria together, collectively, they are going to carry the entire genome of the organism of interest. Our next checkpoint asks, what are the hosts for the genomic libraries described? So we have plasmid libraries that use bacterial hosts, such as E. coli, or we can have phage libraries that use bacteriophage vectors, and these could be examples, lambda phage that we learned about in chapter 6. Lambda phage infect E. coli. So both plasmid libraries or phage libraries are systems that can store recombinant DNA fragments for later screening. In the second question, they ask, what is cDNA? Remember, cDNA is complementary DNA that is synthesized from messenger RNA using that enzyme reverse transcriptase. So this represents only the expressed genes of a cell or tissue. There are no introns or other regulatory regions. So this is commonly used to clone eukaryotic genes into bacteria. But also I put a little note here that we don't really have genomic libraries anymore. Instead, we really use next generation sequencing. So now we have sequencing that is so rapid, we can easily sequence the entire genome of an organism of interest, even humans, within about 24 hours. All right, and that takes us to the end of our first video. In the next video, we're going to finish section 12.1 and talk about transfection. This is how we get recombinant DNA into eukaryotic cells like human cells.